Click for IB Biology, Transcription. Here we're turning the base sequence of the gene into messenger RNA sequence. There's a blank PDF of this presentation down in the description. Annotate that with your own notes if you would like. There's a 10-question quiz, 10 questions, the answers are there of course, and also there's a channel link with an AI prompt to paste into a channel of your own choice, and you can use that to construct individualized multiple choice assessment. So let's take a look at this transcription. So notice immediately that DNA is the double helical structure, the double polynucleotide chain. So this is a double polynucleotide, whereas messenger RNA is a single polynucleotide chain. Another difference is that in DNA we have adenine, thymine, guanine, and cytosine, but in messenger RNA, in all RNA actually, we have adenine, uracil, guanine, and cytosine, and so thymine is replaced with uracil like this. Now up here in the DNA, then we have complementary base pairing of A to T and G to C. In the single chain of messenger RNA, of course, it won't be base pairing as it's just a single chain, but the base ratios will come into action, as we will see as we go through the detail. Now, students of higher level biology, please note that in D2.2, topic 2.2 on the syllabus, there's an entire topic on gene expression which relates to this section here in the model about the initiation of how genes are switched on and off. So you need to study that in conjunction with protein synthesis. A gene begins with a region called a promoter and ends with a region called a terminator. Promoters and terminators are made up of repeating bases like TA, 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 and so forth. So that marks the beginning and the end of our gene. The first part of the gene expression, when the gene is switched on at the promoter region here, is the action of the helicase enzyme that works its way through the DNA molecule, and what it's going to do is to break the hydrogen bonds and open up the two strands, the two polynucleotide strands. And we name those two polynucleotide strands 3' to 5' is called antisense, and 5' to 3' is called the sense strand. This is done with respect to the promoter, which would be in this location here. The antisense strand will act as the template for the building of messenger RNA. So these bases here, which are no longer complementary base pairs, remember these are DNA, they will act as the template upon which we're going to build the messenger RNA. With the gene opened up by the helicase, it's then possible for complementary base pairing to occur between RNA bases of those carrying adenine, guanine, cytosine, and uracil, and they will complementary base pair with the DNA bases in this location here. So the complementary sequences will be A in DNA will complementary base pair to U, not thymine, with U. The DNA, if it has thymine, that would complementary base pair to A in messenger RNA. G in DNA would complementary base pair to C. And C in DNA will complementary base pair to G in messenger RNA. So this is our messenger RNA and this is our DNA, like so. So the nucleotides are going to line up along here using the hydrogen bonding to complementary base pair. And then it's necessary in the process known as elongation to begin making the sugar-phosphate backbone of the messenger RNA. And this occurs in messenger RNA. So important, we understand in messenger RNA, the way in which this occurs is that 5' is added to 3'. This may sound familiar with DNA replication, but here it's the same, except here we're working with RNA nucleotides and not DNA nucleotides. So let's take a look at this process here, which I've enlarged to this. So this is our first RNA nucleotide here. Look at the orientation of that. So the 5' is here like so. And then the second nucleotide comes and complementary base pairs here. So we're going to add the second to the first. So it will be the 5' of the second will add to the 3' of the first. And so the enzyme will make that phosphodiester bond between 5' and 3'. And of course that enzyme which does this is called RNA polymerase. So we're making a polymer of RNA. And you can see this progressed in this diagram here with these phosphodiester bonds which are being added, 5' added to 3', the action of RNA polymerase which ends at the terminator sequence in the gene. At that point, with the messenger RNA complete, it can be released from the template, and then this will then leave the nucleus and go and locate a ribosome so that we can begin the process of translation, the building of the polypeptide. We're first going to take a look at the genetic code itself, which is our next video. There's the link to the channel should you wish to subscribe, and of course the playlist for protein synthesis appearing on the right hand side there.